The aim of this video protocol is to demonstrate the generation and enrichment of neuronal progenitor cells from differentiating neural stem cell progeny using flow cytometry. This is achieved by first isolating and expanding neurostem cells using the Neurosphere assay. The resulting cells are then differentiated through a two-step method called the Neuroblast Assay. Finally, using flow cytometry, distinct populations of cells can be identified and sorted based on their physical and fluorescent properties.
In this protocol, we'll introduce novel methods for both the differentiation of neural stem cells, which is called a Neuroblast assay, as well as for the purification of the resulting immature neuronal progeny using flow cytometry. The Neuroblast assay has a purification phase, which takes about 3 to 4 days, and a differentiation phase that takes about 4 to 5 days. During the differentiation stage, a monolayer of cells forms, which is composed of astrocytes underneath, with clusters of immature neurons on top. When the differentiating neural stem cell progeny are dissociated into single cells and then run through the flow cytometry machine, a sharp difference can be distinguished between the neuronal progenitors and the rest of the cells in terms of their size and internal complexity. Gating the small cell population, which has low forward and side scatter properties, highly enriched neuronal cells can be isolated. To achieve an even more homogeneous neuronal cell population, the dissociated neuroblast assay culture can be stained for PSA and CAM, and the positive cells can be gated and sorted from the small cell population. Okay, let's get started.
Neural stem cells are isolated and expanded from mouse embryonic day 14 ganglionic eminence using the Neurosphere assay, as appears in a separate video protocol which can be found at the address on the screen.